Hello, everyone. Thanks for joining us today. I'm going to talk a little bit about some projects that I've been working on in the lab. My name is Dayana Rivadeneira, and I'm a research assistant professor at the University of Pittsburgh in the Department of Immunology and the Tumor Microenvironment Center. The title of my talk is Fueling the Anti-Tumor Immunity Using Oncolytic Viruses Encoding Metabolic Modulators. And just to set up the stage for my talk, I will briefly just want to touch on the ways that we are currently managing cancer patients with immunotherapy. As you know, in the last decade, checkpoint blocking therapy became the most promising therapy out there, which basically harnesses the immune system as a therapeutic tool. These therapies have been very promising, as you can tell, especially in certain patient populations where you're seeing about 20 to 40% of long-lasting response. Nonetheless, in a lot of these tumor types and other cancers, we are seeing an overall poor therapeutic response. Some of them don't respond at all from the beginning. So there's still quite a bit of space in the field to better understand the mechanisms underlying the lack of response to immunotherapy. But most importantly, I think this therapies, cancer immunotherapy, highlights the incredible potential that harnessing the patient's immune response has against the disease. And so let's talk about what would be the other barriers that T-cell infiltrating into the tumor need to overcome beyond the stimulation of inhibitory receptors, which are the targets for checkpoint blockade. And one of the things that I think it's critical to highlight is that the tumor microenvironment is very heterogeneous among different patients, in which that the tumors can be kind of roughly divided between these hot, cold, and cold ignored tumors, where the T cells have different levels of infiltration within the tumor. And then you'll find that some T cells, or in general lymphocytes, have barriers that have to overcome to infiltrate, such as physical barriers, chemokine signaling, low to no tumor neoantigens, or damage vascularization. And so there's also the recruitment of suppressive immune cells, as well as the chronic stimulation in the tumor that can cause T cell exhaustion. And most importantly, the things that we're most interested in our lab is the metabolic barriers that these lymphocytes have to encounter in the tumor microenvironment. And so focusing on the metabolic barriers that we see in the tumor microenvironment could be considered a metabolic desert where T cells have to compete with tumor cells for nutrients in order to survive. Consequently, T cells find themselves in a very non-permissive microenvironment with very low levels of nutrients, low levels of oxygen, in this case described as hypoxia, as well as the accumulation of detrimental metabolites such as high levels of lactate, which can cause T cells to become metabolically repressed. And these metabolic repression has been seen from our lab in a couple of different tumor types, such as head and neck cancers, as well as melanomas, where you can see that the T cells that infiltrate the tumor, in here described as still, show a decrease in mito-FM, which would be your mitochondrial content, and this is decreased in both head and neck and melanoma compared to your PBL. And so the things that we think about is how to overcome these immunogenic barriers. And so firstly, you want to turn a cold tumor into a hot tumor. So you want to promote that T cell infiltration or lymphocyte infiltration into the tumor bed. And then once you're there, you need to have to utilize metabolic modulators that can improve that T cell function in the tumor microenvironment. And the way that we approach this is to basically be able to combine these two approaches. And so one of the ways that we wanted to address the problem of poor lymphocyte infiltration was with the utilization of oncolytic viruses. And just to give you a brief overview on how they work, oncolytic viruses are replication competent viruses. They come in a few different flavors, as you can see in the graph. And the way that they work is by basically two main mechanisms. They can selectively replicate within the tumor, and what they call oncolysis. And this oncolysis can also produce an induction of an anti-tumor immunity. And one of the major bonuses of these therapeutics is that they can also serve as delivery for vectors for therapeutic genes. You can genetically modify all these genes and include any kind of genetic backpack, basically, that you want to deliver into the tumor. We have already one of these viruses approved by the FDA. It's an HSV-1 engineered virus, and is for the treatment of melanoma. And there are currently several clinical trials ongoing for the combination of oncolytic viruses with checkpoint blockade. And so our lab is particularly using a VACCINIA backbone for our studies, and that's the oncolytics that I will be talking about. So just to get started on our project, we wanted to set up basically a melanoma in vivo model that gives us the makeup that we wanted for this tumor type. Basically, we wanted a tumor that was poorly infiltrated in terms of CD8 T cells, and also that was PD-1 resistant. And the way that we did this was to generate a cell line from a P10BRAF transgenic melanoma model. We had the mice that developed tumors, we isolated the tumors, and developed a single cell clone that we will call clone 24. And as you can see here in the bar graph, in the CD8s, the percentage in the tumor infiltrating lymphocytes is very much decreased compared to your typical B16 infiltration CD8 population. And on the right, you see the tumor growth graph, where you can see that the tumors that were treated with anti-PD-1 have no response to the treatment. So we have a melanoma model set up for investigating our therapeutic. And so here, initially, what you can see is that when we treat melanoma tumors, this clone 24, with a wild-type VACCINIA virus, you are starting to see some responses for the tumor. You see a partial response of about 42%. And so we wanted to see what was actually the makeup of the infiltration of these tumors after treatment. And as you can see here from single cell analysis, we can start seeing how are these lymphocytes distributed in the tumor. And one of the important things that here to highlight is how in the PBS group alone, you have very little T cell infiltration. As you can see by the pie chart, it's about only like 7%. There's quite a bit of macrophages and monocytes. And then particularly when you treat the tumors with the VACCINIA virus, there's an expansion of the T cell population. There's also an expansion of monocytes and a decrease in the overall macrophage population. So this gives you an overview of how the VACCINIA virus affects the tumor infiltrate after treatment. And so looking a little bit more specifically at the tumor infiltration by flow cytometry, what we can find by flow cytometry as well is that compared to the lymph node, where you see no changes in CD8 populations with the VACCINIA treatment, which is also very nice to see that there's no effect into the lymph nodes, you see that in the TIL, there's a dramatic increase of the CD8 population after the VACCINIA treatment. And also important to highlight is that you see a decrease of the FOXP3 positive Tregs population after the VACCINIA treatment. And this is something that I will touch on towards the end of the presentation. Another important characteristic that we found in the CD8 population of these tumors was that there was a deficiency in their metabolic state, where you can see here by TwinBDG that shows glucose uptake and MyoTracker for mitochondrial content, we see an overall decrease on these two markers, showing that there are metabolic deficiencies in the T cell infiltration. And so the idea now moving forward is how can you restore that metabolic function of the T cells in the tumor microenvironment. You got the T cells into the tumor by using the oncolytic. Now you need to overcome the different barriers they encounter in the tumor. And so our lab has been able to do this in a couple of different ways throughout the years. And as you can see here from this text, we've done this by inducing PD-L1 alpha expression, which promotes mitochondrial biogenesis. We've done this by reducing oxygen consumption from tumors through the treatment of metformin, allowing T cells to use oxalots. And we've done this also by using 4-1BB agonist, where we know that it promotes mitochondrial function and consequently improving checkpoint blockade. And so we were looking at other metabolic players that can also help us improve T cell function. And we decided to look a little bit into pro-inflammatory cytokines. And what we found was that from the literature is that leptin can also push T cells towards a more oxidative state, where there have been studies that shown that they can stimulate fatty acid oxidation as well as drive glucose to fatty acid functions. And so this gave us an idea that leptin could be an interesting player. And most importantly, that I'm not showing here, we do see that exhausted T cells can express highly the leptin receptor. And so to do our initial studies, we did a CD8 T cell culture in vitro, where we treat the T cells with different concentrations of leptin. And what we find is that through metabolic analysis of seahorse, we show that leptin can improve the oxygen consumption rate, as you see in the top row of graphs. There's a higher OCAR, which is oxygen consumption rates in the basal state, as well as higher spare respiratory capacity. And we can also see that there's no major changes in terms of glycolytic capacity, shown by the ECAR plot. And then when we look specifically at by flow cytometry and mitochondrial content and glucose uptake, we do also see that by leptin treatment, you have an increase in the mitochondrial mass staining, showing that leptin can actually have a pro-oxidative effect on T cells. And so how can we test the effect of leptin directly in the tumor microenvironment? The way that we came about to do this experiment, we decided to take our melanoma model and transduce them with a leptin gene, where we can see if the tumor will produce leptin, release it into the microenvironment. And this is exactly what we find. When the leptin is expressed by a tumor, they actually release it into the tumor microenvironment. And when you put these tumors into mice and follow their tumor growth, we find that the tumors that are expressing leptin have a much controlled tumor growth compared to their control counterparts, and also increase the overall survival of the mice. When we look at the tumor infiltrating lymphocytes directly, we do also see, similar to the oncolytic treatments, we see an increase in the infiltration of CD8 as well as the infiltration of CD4 T cells. Most importantly, we wanted to assess if this effect of leptin on the tumor growth was directly correlated to the effect of the CD8 T cells in the tumor. So the way that we did this was to deplete the mouse from CD8s through using an antibody. And what you find is that when you have a tumor that is expressing leptin, but you depleted the CD8s from the tumor, you no longer have that controlled tumor growth, and these tumors start growing the same as controls, showing that this effect of tumor growth is dependent on CD8 function. And further, we were able to show that this tumor control growth was also dependent on the expression of the leptin receptor on the T cells, where in this experiment we have leptin receptor flox mice with a CD4-CRE, and we see that the tumors that were expressing leptin that were injected into the leptin receptor knockouts also they lose that controlled tumor response, showing overall that the effects that we're seeing in tumor growth are dependent on the tumor infiltrating lymphocytes and these lymphocytes being able to express the leptin receptor. And so what is the functional state of these tumor infiltrating lymphocytes? We find that these tumor infiltrating lymphocytes, when they are expressing leptin, they have a better functional capacity, as you can see here by staining, by looking at the expression of interferon gamma and TNF alpha. And as well, that we are observing an improvement in mitochondrial function, where we're seeing a higher mitochondrial mass in the tumors that are expressing leptin. And so moving forward, the idea is to be able to deliver the leptin directly into the tumor microenvironment. And the way that we did this was by cloning the leptin gene into a VACCINIA viral expressing virus. And in that way, what we do is that we deliver the VACCINIA to the tumor, the tumor the virus will release the leptin into the tumor microenvironment, and this will improve the T cell function once they get into the tumor. And when we're looking at the therapeutic responses of this vaccinia virus, what we see is that the clone 24 melanoma model, we see a much better response with the virus that expresses leptin. As you can see here, there's a complete response of about 27% in the melanoma model. We also included a pancreatic cancer model, PANCOS-2, where you don't see a complete response, but you definitely see a much better partial response with the leptin-expressing virus compared to the virus control. When we're looking at the tumor-infiltrating lymphocytes, we also see that these tumors have a much better functional T cell infiltration, where you can see that they're expressing much more cytokines in terms of TNF alpha and interferon gamma compared to the control virus. They're also generating a lot more mitochondrial mass, and this is observed by the staining of VDAC. You're seeing a lot more VDAC, a higher percentage of VDAC in the leptin-expressing virus tumors compared to their controls. And importantly, too, we are actually observing an increase in proliferative capacity in these T cells as well, where we're seeing a higher percentage of Ki67 staining in the T cells from the tumors treated with the leptin vaccinia virus. And so just to look a little bit further at the populations that are in the tumor after treatment, and looking specifically at the populations in terms of like the T cell clusters, we can divide the T cells that are infiltrating into these tumors after treatment in different populations, such as memory, cytotoxic, CD4 T cells, Tregs, exhausted. And one of the things that it was highlighted during the analysis was that there was an increase in general of the memory population. And so when we did TIL analysis, we did see that the tumors that were treated with the vaccinia leptin have an increase in CD127 expressing tumor T cells, showing that in general there's a skewing towards a memory population, which would be also beneficial for better response to the immunotherapy. So trying to see if this memory activity is true, what we did was a memory experiment where we had a cohort of mice that got treated with the vaccinia leptin expressing virus. As I mentioned before, there's a complete response of about 30%. So we took all of these complete responders and we kept them alive for a few days, up to 20 days, to make sure that they don't regress. Once we knew that the tumor was completely eradicated, we took this cohort, including a group of mice, wild-type mice, injected tumors again to these mice. And what we find is that out of the 11 complete responders, 8 never had tumor regression. They never grew another tumor again. Only 3 had some tumor grow again. And even then, it was a lot slower than the controls that all developed tumors. So this also shows that by improving the metabolic capacity of the T cells in the tumor microenvironment, you're also skewing them towards a more memory-like population and probably potentially having a better response to therapy. And so just to give a brief overview on what I'm showing you now, we can say that our data suggests that leptin can enable anti-tumor immune responses in a tumor microenvironment that is metabolically deficient, and that oncolytic viruses expressing leptin can enhance the overall therapeutic response to oncolytics, and that using these two strategies, of inducing T cell infiltration and at the same time targeting the metabolic deficiencies encountered in the tumor microenvironment could be the most appropriate way to go after a better therapeutic response. And so for future directions, we're looking into determining the effects that the oncolytic virus has directly on the TIL population. This is an ongoing project right now in the lab. And of course, exploring the metabolic regulators, other metabolic regulators that can be expressed in oncolytic viruses in order to improve therapeutic response. So we're constantly thinking about other ways that we can improve oncolytics and always looking for targets, metabolic targets. And just to give you a little bit of a brief data that we have that is preliminary, like I mentioned before, I talked about how FOXP3 CD4 positive T cells population decreased after VACCINIA treatment. So that we found it was very interesting that in a way we're targeting the Tregs, which is good in the terms of like a tumor setting. And so in collaboration with the Hink lab here at Pittsburgh, who develops these TGF-beta inhibitors, he produced a TGF-beta mini monomer, which is basically a monomer that binds to the TGF-beta R2 receptor. In this way, it outcompetes a TGF-beta, and that way there's no TGF-beta downstream signaling. So what we initially did was treat cells with the VACCINIA virus and get the supernatant from VACCINIA control as well as the VACCINIA that had the TGF-beta monomer. And what you see here in the graph, when you just have the control VACCINIA plus TGF-beta, you have a higher TGF-beta activity. When you include the supernatant from the cells that were treated with the VACCINIA TGF-beta virus, you can see a decrease in TGF-beta activity, and this activity gets diluted as the supernatant gets diluted as well, showing that it's specific for TGF-beta activity. And most importantly, when we look at the tumor growth with this virus, you actually do see an improvement in treatment compared to the VACCINIA control with the TGF-beta virus. And so this is just the preliminary data that we've been developing in the lab, and we're further exploring with different compounds. And so just to give a overall summary for my talk, VACCINIA can be utilized to induce an immune response in cold tumors pretty efficiently. And this immune response needs some metabolic support to be able to function better. And most importantly, VACCINIA viruses, as well as other oncolytics, can be genetically modified to provide that metabolic support. And so I just wanted to give a quick acknowledgement for my lab that I'm in with my mentor, Greg Dalgoff, and everyone in the lab, and of course my collaborators at Pitt that have been instrumental for this project to move forward. And with that, I'm done. And if anybody has any questions in the future, please contact me. I'll be happy to answer any questions you guys have. Thank you.